Let us continue our discussion on human health and disease. In this video, we will talk about immunity, and this is part A, which means that we will discuss about the innate immunity in this video and some parts of the acquired or adaptive immunity. The remaining parts of the topic will be dealt in the next video. So straight away coming to question number 1, what is immunity? It is the ability to fight disease causing organisms. What are these disease causing organisms called? So they are called as pathogen. They are called as pathogen, and pathogen infect a susceptible host. So they infect a susceptible host, and if the host is trying to fight it, that would be called as its immunity. And every human being, if we are talking about the human beings, will be capable of mounting an immune response against the pathogens that are trying to infect his or her body. Now this immunity is of two types. Basically it is of two categories or varieties. One would be called as an innate immune response. So this is what we are going to discuss first in this video. So innate immune response or innate immunity. Now the first question to understand about innate immune response is that it is a non-specific response. So point number 1 would be non-specific. So unlike the adaptive immunity, so just to give you an example, in adaptive immunity, the type of antibody molecules that will be acting, say for example, against a particular bacteria, say for example Salmonella typhi, they will not be acting against other bacteria at all. It is very specific. It is very specific. Against a particular pathogen, a particular type of antibody molecules are formed, and there is a very specificity between these things. But here the response will be against a large number of different categories of microbes at the same time. So we are saying this is a non-specific immune response. The second is, yes, that it is present since birth. So it is present since birth, no doubt about it. So I hope both these points are now absolutely clear. This can be a good question in your NEET entrance exam. The third thing is that this innate immune response is because of certain barriers. And what are these barriers? What they do, they actually try to prevent the entry of the pathogen into the body, and if at all it enters the body, immediately try to confront the pathogen and eliminate them very quickly. So this is what these innate immune responses, which are in the form of barriers, actually do. And what are the barriers that we know? Number 1 barrier would be the anatomical barrier. So the structural barrier, the anatomical barrier are one of the barriers which are the part of innate immune response. So you know that your body is covered on the external surface by the skin, and internally, if you think about your GIT, that would be very easy to understand. There are mucous membranes. So we are talking about the covering of the body in the form of skin and mucous membrane, and they are protective covering. Mucous membrane also are lined by mucus that can actually stick the pathogen on its surface and therefore prevent its entry into the pathogen. So this skin and mucous membrane is the first line of defense. So skin is now regarded as an organ. So this is the first line of defense against, now look at these words, against the entry of pathogen into the body. So against the entry of pathogen, this becomes the first line of defense. At the first instance, they are not going to allow the pathogens to enter in your body, and therefore just understand this, that they will be preventing all sorts of microbes to enter into the body, not very specific ones, and therefore you can easily understand that this response is actually non-specific in nature. Any breach in continuity of the skin and mucous membrane, which is called as an ulcer, will be becoming a portal of entry through which these pathogens can enter into the body, and then they actually infect the body by either multiplying, growing, or by performing both the functions inside the susceptible host body. So this is barrier number 1, anatomical barriers. The second type of barriers are the physiological barriers. So the responses to these pathogens that your body is likely to mount, they are called as the physiological barriers. Mind you, I told you that innate immunity is because of certain types of barrier. We are looking at the types of barrier. Number one is anatomical, the second is physiological barriers. A very, very important physiological barrier is the presence of HCl hydrochloric acid in your stomach. It is a very, very potent bactericidal. So it is a very, very potent bactericidal. It can kill large number of bacteria. Do you know that there is a bacterium which is called as Helicobacter pylori, and that Helicobacter pylori is responsible for large number of duodenal ulcer cases, and it is an acid tolerant bacteria. Do you know what do you know something about lactic acid bacillus? And otherwise large number of pathogen bacteria can actually be killed by the HCl that is present in the stomach, and it's a very, very important barrier for these bacteria. The second thing, as you know, is a lysozyme. So this lysozyme is present in body secretions. It may be present, in saliva. It may be present in tear, for example. And this is again an enzyme that is actually bactericidal because it disrupts or digests the cell wall of the bacteria. And if the cell wall of the bacteria is removed, the bacteria will not be able to survive in a hypotonic environment. The fluid will enter the bacterial cell and it will burst. So this is again a very important physiological barrier in innate immune response. One more thing that I can add to NCRT is actually fever. So fever is a physiological response against infection. The body is trying to increase the temperature so that it does not remain conducive for the growth of the microbe. So it is trying to control the microbe. That is the reason that why we normally do not treat low grade fevers very early. So that means a person may be having 100 degree Fahrenheit. So doctor normally advises that unless and until the fever is high, do not take antipyretic. Incidentally, antipyretic is a drug that is used to decrease the fever or decrease the temperature in a febrile patient. And this fever, the low grade fever in initial stages, may actually be helping you fight the disease. Okay, if it becomes very, very high, then it becomes very detrimental, and then the problems are more rather than the advantages, and then we try to bring the fever down. So I hope now this type of thing is also available. The third barriers that are present in innate immune response are called as the cellular barriers. Now this is very important. Some important questions can be formed here. So we are looking at phagocytes. So you have read about WBC, and you know that there are certain cells which are phagocytes, and these includes the neutrophils, and neutrophils, if you read your NCRT, are also called as polymorphonuclear lymphocytes. So this is polymorphonuclear leukocytes. Polymorphonuclear leukocyte, I have already explained to you in the chapter on human physiology that they have 2 to 7 lobed of nucleus. They are actually granulocytes. And the other cell can be a monocyte. Now monocyte is the largest WBC. Monocyte comes out, squeezes out of the capillaries and goes into the tissues, gets differentiated into macrophages, and then the macrophages are very, very potent phagocytes. So we are talking about neutrophils, we are talking about monocytes, but monocytes, they squeeze out of the capillaries, reach the tissues, get differentiated into macrophage, and once they are differentiated, macrophages are much more potent phagocytes than neutrophils. Neutrophils do not need to differentiate, they are ready-made phagocytes, but the phagocytic capability of macrophage is many folds than that of the neutrophils. I hope these things are absolutely clear. And these macrophages, if you remember the diagram of your class NCRT, the areolar tissue, so these macrophages are located in the areolar tissue, and if you remember that areolar tissue is beneath the skin, and as I told you that any breach in continuity of the skin or the mucous membrane will become a portal of the entry to the pathogen, but there itself there would be pre-formed macrophages. As soon as the first line of defense against the entry of the pathogen is breached and the pathogen enters into the body, then another line of defense is available, which are these macrophages, the already differentiated cells that are macrophages that are present in the areolar tissue. They will immediately immediately start phagocytizing these bacterial cells. Macrophages are also advantageous located in many locations of the body, just going a little bit outside NCRT. So that means the macrophages are present in the alveoli, again a portal of entry for the pathogen. So they are present in alveoli, they are present in the blood spaces or the sinusoid lining of the liver, and there they are called as Kupffer cells, and they are also present in brain where they are called as microglia, also present in spleen and the lymphoid tissue, and they are called as reticulum cells. So we are looking at macrophages in areolar tissue, macrophages in alveoli, macrophages in hepatic sinusoids, in the lymphoid tissue, and also in the brain, the neuroglial cells called as microglia. So they are guarding virtually every possible portal of entry of the pathogen into the body, and as soon as the pathogen enters, these are the first cells that confront these pathogens. I hope these things are now absolutely clear. The second cell that we are talking about here are the NK cells. Now these NK cells, they are called as natural killer cells. So they are natural killer cells. Now these natural killer cells are analogous to T lymphocytes. Mind my word, they are analogous to T lymphocytes. That means they are cytotoxic. So the natural killer cells are cytotoxic, very important, very important in innate immune response. They are, I am saying, analogous. They are analogous to T lymphocytes. So they are not T lymphocytes, they are analogous to T lymphocytes. I have already mentioned that they are also cytotoxic. Present in them are certain chemicals. So say for example, they may have perforins and have granzyme. So that means this is again going little bit outside NCRT. They may contain perforins and they contain granzyme. So when these things are released in the vicinity of the pathogen, etc., or what can happen is that they can form channels, and the water can enter into the cell and leads to the death of the cell. Similarly, they are very effective against virus. They are very effective against virus. They are very effective against virus, and they are also effective against the tumor cells. So again, looking at the analogous nature to the T lymphocyte. Now what happens is that suppose a cell has been infected by virus and perforins and granzyme have been released, because you want that this virus should not spread, and therefore what happens is that they will create what is called as some of the pores because of these perforins. So something wrong has happened. So let me make the diagram again. So this is a cell. This cell has been infected by the virus, and because of these perforins, some perforated channels have been formed, and because of this, the water will enter, and this will lead to the bursting of this cell. The cell has been affected by the virus, and therefore the virus will not be able to multiply in sufficient number because the cell is dead. And similarly, the granzyme will induce apoptosis here, and this will lead to the early death of the cell, and again decreasing the viral load. These are the mechanisms by which natural killer cells can actually act, and I hope now you understand about the cellular barrier. Some important questions can be asked from this section of immunity. The final barriers, type of barriers that are present in your body would be the cytokine barriers, the immunological barriers. The cytokine barriers, we are talking about the immunological barriers. That means immunomodulators we are talking about. Everything is actually immunological. So we are talking about immunomodulator. So cytos is a cell, kine is a chemical. So any chemical that is secreted by a cell that actually has an effect on the immune system of the body would be called as cytokine. If such thing is actually secreted by a lymphocyte, it would be called as a lymphokine. And some examples of the cytokine can be interleukins. And one of the example of cytokine would be interferon. So these interferons are glycoproteins, and they are secreted by virus infected cells. So this is a very important thing. So they are secreted by virus infected cells. This question has been asked many times. One, interferon is a glycoprotein. It is secreted by the cells that have already been infected by the virus, and in the best of the scenarios, when they are secreted, they actually heightened the immune response of the adjacent cells against the virus, and therefore help control the viral infections. So if the body is having a viral infection, it is likely that the level of the interferons will be increased. So again, I am telling you the scenario when the virus infected cell releases the interferon, it actually heightens the immune response of the adjacent cell, and this is how the body is able to fight a large number of viral infections. Interferons also interfere with the replication of the viruses, and that is why in the first place they were actually called as interferons. So interferon word comes from their capability of interfering with the virus replication, thereby reducing the viral load, and then the body can take care of the virus adequately. So these are the 4 types of barriers that are present in the innate immunity. So mind you, it is a non-specific type of immunity. It is present since birth, and it is mediated through certain types of barriers, and we have done a lot of questions. I hope you remember all of them. Now moving on to the acquired immunity. So moving on to the acquired immunity. So the acquired immunity may also be called as an adaptive immunity. So this is an adaptive immunity. This immune response, unlike the innate immune response, is highly specific. So highly specific. So this is a very, very important thing. The type of the response against Mycobacterium tuberculosis will be specific to Mycobacterium tuberculosis. It will not work even against Mycobacterium leprae. So Mycobacterium tuberculosis causes tuberculosis, Mycobacterium leprae causes leprosy. Both are from the same genus, but what I am saying, the specificity is such that the response that will be against Mycobacterium tuberculosis will not be effective against the response to Mycobacterium leprae. I hope now the first point is absolutely clear, and as you can see that it is acquired during the lifetime, so it is not present since birth, and that is the reason it is called as acquired immunity. Normally, as I told you, a very, very important word that can replace the term acquired immunity would be adaptive immunity. So try to remember adaptive immunity. Now this is a function of lymphocytes. So adaptive immunity, so I may be using the word adaptive immunity obviously because that is a part of my habit, but adaptive immunity is acquired immunity. So it is a function of lymphocytes, and like all other blood cells, the lymphocytes are also formed initially, at least in the bone marrow. So we know that all blood cells are formed in the bone marrow. Lymphocytes initially again are formed in the bone marrow. Later on they can actually divide and form new lymphocytes in the peripheral lymphoid organs or the secondary lymphoid organs, but to begin with they are also formed in the bone marrow. Now what happens is this is an important flow chart that I am going to make here. Please try to understand that the bone marrow, when it starts producing lymphocytes, this will be occurring in the perinatal time. So this is around birth, just a few months, just a little bit earlier than the actual birth happens. This thing is getting started, and this will continue after the birth and later on develop during the lifetime. So therefore, when the newborn is born, is not having a lot of adaptive immunity, and you know the importance of colostrums that we have already talked about in the chapter in human reproduction. You know about the importance of this breastfeeding for the newborn infant. You already know that it has IgA class of antibodies, the infective antibodies, and they can be critical for the survival of the infant. And the bone marrow, actually, when it synthesizes lymphocytes, some of these actually are transported to a gland which is called as thymus. So we already know by the discussion on human physiology endocrine system that thymus is a gland that is present ventral to the heart. So it is present ventral to the heart. You also know that it atrophies with the age, and in the thymus some of these lymphocytes will become processed. So they will become processed. We will understand later what this processing means. And then it comes to circulation, and these processed lymphocytes, they go and rest in the secondary lymphoid organs. So we are saying these are the processed lymphocytes. They are called as T lymphocytes. They are called as T lymphocytes because this processing has happened in thymus. So this T stands for thymus. So this is now very easy. Lymphocytes, some of them go to thymus for processing. After processing, they are resting in the secondary lymphoid organ. Now some of the lymphocytes in human beings, they remain in bone marrow and they get processed in the bone marrow itself. So they get processed in the bone marrow. Again, the processed cells come to circulation and they go and rest in the secondary lymphoid organs. So this is the secondary lymphoid organ. Say for example, it may be spleen, it may be lymph nodes. So these are the secondary lymphoid organs, the tonsils, etc. So they all are secondary lymphoid organs, the mucosa associated lymphoid tissue, and there they are resting. From here they will respond to the pathogen. I will come to this point in a minute. So these type of cells will appropriately be called as B lymphocytes, though sometimes you must remember that these types of lymphocytes, the site of processing was actually discovered in birds, and birds also begin with B, and they have an area which is called as bursa of fabricus, which incidentally also begins with B, and bone marrow also begins with B. So for us in human beings, there is no bursa of fabricus. We are not birds, we do not fly away. We are saying that these B lymphocytes actually are processed in human beings in the bone marrow. That is the business end of the thing that I am talking about. This is likely to be asked in the entrance, and then they are called as appropriately B lymphocyte. Now there are two organs. These are called as primary lymphoid organs where the processing takes place, and these are the bone marrow and thymus. So the bone marrow and thymus are called as primary lymphoid organs. So they are called as primary lymphoid organs because the processing takes place here. Now this processing means that they are a sort of introduced to all the self antigens that the body can produce, and they are asked, if I may say so, not to respond against them. So this is what happens. It is during processing that the immune cells of your body are able to identify self antigen. So they get processed only to act against the non-self antigen. And a very important property of adaptive immunity is the ability to distinguish between self and non-self. So that processing is happening. So they are getting ready for the function in the primary lymphoid organs. But when the pathogen enters into your body, and then these cells will actually respond from the secondary lymphoid organ. This is a very, very important question that is given in NCERT. So the environment for the development and the processing is provided by the primary lymphoid organs, and the actual interaction with the pathogen and the antigen actually takes place in the secondary lymphoid organ. I hope these things are now absolutely clear. So now we know about what are T lymphocytes, what are B lymphocytes. I hope this thing is now absolutely clear. Now, there is a term that I am using again and again which is called as antigen. So antigen is a chemical. Antigen is a chemical. It can be commonly protein or carbohydrate. You know that blood group antigens are carbohydrates, and that is very unique. So there is a disease causing organism which is called as pathogen. On its surface may be present an antigen. This antigen would have a unique chemistry. So when the pathogen enters your body, this antigen is unique compound. It is not present in the human body, and this is how the immune cells are able to identify that this is alien or foreign, and it is non-self, and they start responding against it. So it is the entry of the antigen. And it is the antigen actually that initiates the immune response in the host. This is a very important question. So that means it is the antigen that initiates the immune response in the host. This is a very, very important question. Please try to differentiate again between pathogen and antigen. And this antigen, what is a self antigen? Self antigen would be present on the surface of our cells as well. Do you already know about the presence of the carbohydrates that are present on the outer surface of the cell membrane. You already know from my discussion on cell cycle and cell division that they are involved in cell to cell recognition, and you already know about the blood group antigen. That is the most easy thing that you can actually recall right now. So these antigens are present on the surface of RBC, but they are self antigen, and the immune system actually responds against the non-self antigen. I hope this is now absolutely clear. Now what happens is that once an antigen is actually identified by the immune cell. So this is the secondary lymphoid organ. Now I am telling you again that this is the area, the secondary lymphoid organ, where the interaction between the antigen and the antibody molecule will actually take place. So there would be very specific T lymphocytes. So in the previous slide I said that T lymphocytes, differentiated T lymphocytes are present here. This time I am talking about different colonies, T1, T2 and T3. Similarly, there will be different B lymphocytes, B1, B2 and B3. So these are the processed T and B cells that are residing in the secondary lymphoid organ, and it is and one of them actually identifies the antigen. The specificity is such that this antigen will be identified either by the T cell or by the B cell, not both. Again, look at the specificity of the immune response, and even in T cell there would be a subset that is going to identify it. Not every T cell will be identifying it, and therefore what happens is, say for example, if it is identified by, say for example, B1, and then what happens is immediately on identification of the antigen, the B1 undergoes cloning, so it makes a large number of its own type of cells. So these are all B1 cells. So this step is called as clonal selection. So the term is self-explanatory. This is called as clonal selection. So immediately the antigen is identified by the B1. B1 actually undergoes a propagation. It becomes a clonal selection. And what happens is that out of these, some of them will actually get converted into what are called as memory cells. So these are called as memory cells, and they will go back and reside in the secondary lymphoid organ. So this is a very important property of adaptive immunity. It retains the memory of all the encounters that it had already had with the antigen and makes memory. So that means the number of the cells that are now present, the type of B1 cells that are present in this secondary lymphoid organ are not one but are many. So next time this pathogen or with antigen actually enters your body, the response will be huge, and this is how large number of diseases, they are actually sometimes associated with very long term immunity. You know about many viral infections. So this is because of the formation of what are called as memory cells, and this principle we will use in vaccination as well. Okay, how this memory cell and the secondary response to the antigen would always be anamnestic. So the secondary response would be anamnestic. So we are talking about some very important properties of the adaptive immunity, the ability to distinguish between self and non-self, obviously the specificity part of it, and then we are saying that the secondary response, the formation of memory, and this is causing the anamnestic response. The first time the pathogen or the antigen enters the body, the response will be primary. It will be a limited response. It may take time to develop, but the second time the same antigen enters into the body, the response will be anamnestic. It will be much higher and will be very rapid as well, and will be able to take control normally of the infection in any other subsequent exposure to the same antigen for the immune cells of your body. So this is what happens. The other B cells, they get differentiated into effector cells. They get differentiated into effector cells, and these effector cells are actually called as plasma cells, and there may be some time that may be required for the differentiation of these cells into plasma cell. But once plasma cells get differentiated, they secrete large number of antibody molecules into the body fluids. So it is the plasma cell which is the effector B cell. It secretes large number of antibody molecules into the body fluids, and because the body fluid are called as humor, this type of immunity is actually called as humoral. So a type of adaptive immunity which is called as humoral immunity, because these antibodies are present in the body fluids. Remember cardiovascular system, class 11th physiology, you know that gamma globulins are immunoglobulins antibodies, and they are a type of plasma proteins. Now what happens in the case that this is actually identified by the T cell? So let us say that T1 identifies it. Again there will be clonal selection. Large number of cells will be formed, and again what is going to happen, that some of them will be converted into the memory cell, so they also retain the memory, no doubt about it. So there will be formation of memory cell. Again the response would be anamnestic for the subsequent exposure to the antigen, and then they will be differentiated into the effector cell, which are called as T cytotoxic cell. But these T cytotoxic cell actually do not secrete any antibodies. They directly act against the pathogen or the antigen, and because this is a cell acting against a pathogen or antigen, so appropriately this type of immunity is called as cell-mediated immunity. And cell-mediated immunity is an important question for your NEET entrance exam, because this is the type of immunity that is actually effective against virus, that is effective against the tumors, the cancers, and that is also, this is an important question, that also responsible for graft rejection. So it is also responsible for graft rejection. This question has been asked multiple times. So this is an important question. They are also responsible for graft rejection. I hope all these things are now absolutely clear. That does not mean that humoral immunity is less important. It is very effective against rapid going bacteria, and such infections are actually very, very commonplace. So both are equally important. So I hope now you understand that what exactly is a humoral immunity and what is a cell-mediated immunity. If you want to understand the details, so I have given a very thorough explanation of the antigen presenting cells, the T cytotoxic cells, the T helper cells, the T suppressor cells, all these things, what are the interleukins that are being secreted, what is the central role played by the T helper cell in the immune response for adequate adaptive as well as innate immunity in my video in the normal course. And because this is our final revision before the NEET entrance exam, so we are limiting ourselves largely to the concepts which are given in NCERT, from where it is expected that almost all questions will be based, and therefore I am not going into these details. If you are interested, you can actually always refer to the video that I have done in the main course. Meanwhile, in this chapter, let me discuss one more thing, that these plasma cells, they secrete antibody molecules. And what are these antibody molecules? So you know about them, that they are actually gamma globulins, they are immunoglobulins. So that means, if you remember from the blood, the discussion on the blood chapter, you know that they are immunoglobulins, and immunoglobulins will be abbreviated as IgGs, and the monovalent form. Now look at this. Again we are doing some important questions very rapidly, and just try to pay your attention. If there is a problem, stop the video, look at the NCERT and come back and watch the video. We are saying that it has the monovalent form of an antibody molecule has 2 heavy chains and 2 light chains. So there are 4 polypeptide chains. This is a protein we are talking about. The monovalent form of the antibody molecule is having 2 heavy and 2 light chains, and they are held together by, this is an important question again, so they are held together by what are called as disulfide bonds. So we know that disulfide bonds happen when cysteine amino acids come adjacent to each other, and though methionine is also sulfur containing amino acid, but it is not involved in disulfide linkage. So this is the we would say a cartoon of an immunoglobulin molecule monovalent form. What I will ask you is to stop the video right now, open the diagram in your NCERT and see whether in this diagram there are written two letters N and C, and just identify which is the N end and which is the C end of a polypeptide chain. Because if you remember biomolecules, you know that when amino acids will be joined end to end by a peptide bond, and this primary structure would have an N terminus and a C terminus. This is the amine group, this is the carboxyl group of the amino acid. So just look at the NCERT, stop the video, watch it. Many of you might not have noticed that there is an N terminus and a C terminus mentioned there. Please look at it, and then we can continue with the discussion. Now in this antibody molecule, so this is the region, a sort of half of the region of the lighter chain and 1/4 of the region of the heavier chain. As you can see, this region is called as the variable portion. So this is called as the variable region of the antibody, and this one is called as the constant region of the antibody. And this obviously is called as variable, therefore it has large number of variations present in it, and it is a hot spot of mutation. So the genes that are responsible for the formation of antibody molecules, you can understand, because antibodies are proteins, so there will be genes of which they are expression, and you know that we can make a huge number of types of antibodies, and actually much more than the total number of genes, and all genes in your genome are not involved in antibody formation. So how come a small number of genes actually is producing large number of diverse antibody molecules? So it is because of this variable portion or this variable region, because there is large number of variation in the amino acid residues that are present here because of the gene being a hot spot of mutation. There are what are called as VDJ recombinations. Do not go into the unnecessary detail, but just keep the idea in the mind that a few hundred genes actually will be making hundreds and thousands and thousands of types of antibody molecules, and that can be explained by this thing. Now present between the heavy and the light chain in this variable region is what is called as the antigen binding site. So this is what is called as an antigen binding site, and this is where the antigen actually binds to the antibody molecule, and this thing is called as clumping or agglutination. That is the main mechanism of action of an antibody molecule. So antibodies most commonly agglutinate or clump the antigen. You must be aware of this thing. We have already discussed this type of thing in blood group antigens, that blood typing test. I have given you the idea of agglutination, and the antibodies can actually neutralize the pathogens. They can actually precipitate out the toxins, and they can actually act as opsins. They are involved in opsonization, which means that they stick to the surface of the pathogen, so that the phagocytic cells etc. are easily identifying these type of pathogens, that these are the cells which are to be phagocytized. So a sort of sticker, you can assume, they get attached to the surface of these foreign cells, so that your immune cells are actually able to identify that this is the cell that needs to be phagocytized and kill, and the response is becoming activated. In higher classes you will know something about a complement system. It is a cascade of proteins, and antibody molecules can actually activate the complement system as well. So there are many methods by which antibody molecules, they can actually cause lysis as well. Directly they can kill the cell. So there are the various mechanisms, plus please try to remember agglutination and opsonization. And a monovalent antibody has two antigen binding sites. There are various classes of antibody molecules that are present, and they include what are called as IgA. So you already know about IgA class of antibodies. It is a divalent antibody. So divalent antibody will be something like this, and you can imagine another monovalent form present here, and will have four antigen binding sites in total. It is also called as secretory antibody. It is also called as anti-infective antibody, and it is present in the body secretions like saliva and colostrums, if you remember. So this is IgA class. The question has been asked on this. The second that you need to remember would be IgE. Now IgE class of antibodies we will see later on, they are involved in allergic responses. So we will come back to it in the next video, that what are allergies and what are IgE class of antibodies. The next thing that you must know is about IgM. This IgM is the heaviest antibody, it is pentavalent. So it is pentavalent, it is heaviest. Now heaviest antibody will be something like this. So heaviest antibody will be something like this. So we are saying you are making the structure pentavalent 3, 4 and let us say 5. So something like this. You can imagine this is a pentavalent antibody molecule, it is heaviest, and because it has large number of antigen binding sites, so generally in the primary response this is the class of antibody that is secreted in the body. So you can understand the primary response is not a very huge response, it is limited in scale. So why not secrete antibodies which can actually agglutinate large number of antigens, because this is the heaviest one, it is pentavalent, it has 10 antigen binding sites, so it will be able to agglutinate more. So you can compensate for the number by increasing these antigen binding sites, and it has a diagnostic relevance as well. If a person is infected by a particular organism for the first time and you do an ELISA, the ELISA tells you that the type of antibodies that are present are IgM. So you know that it is the primary infection. This has happened for the first time. So they are involved in the primary responses. The most likely question from the class of antibodies that you are going to be asked, apart from obviously IgA, would be about IgG. So they are the most common type of circulating antibodies. Around 70-75% of all circulating bodies in the human beings are actually of IgG class, and they are able to cross placenta. So they are most common, they are able to cross placenta, and I mentioned earlier, they are also able to activate the complement system and are the class of the antibodies that are generally released in large number in the anamnestic or the secondary responses. And therefore if the test of ELISA that we were talking about comes back positive with IgG antibodies, you know that this is a secondary response rather than a primary response. So this is an important thing that we have talked about, what is an antibody molecule, and now you have a fair idea of acquired or adaptive immunity. In the part B of the video, we will be looking at the remaining topics from the immunity section of human health and disease.